In this video, I will be discussing Nirenberg and Matte's experiment and Nirenberg and Leder's experiment. In Nirenberg and Matte's experiment, they were looking to break the genetic code. For this experiment, they chose to use a cell-free environment, which is basically just a cytoplasm of cells, everything within the cell minus the walls. They used E. coli bacterial cells for this. They also used 20 test tubes that contained amino acids and one of the amino acids which were radioactively labeled. They then proceeded to add polyuracil, a homopolymer, to each of the test tubes in order to see if any unusual activity occurs. And unusual activity did happen when they added the poly-U to the hot tube, which the hot tube is basically the tube that had the radioactively labeled amino acid. The homopolymer, they had discovered that the homopolymer, which is the mRNA poly-U, had destructed the chain of amino acids to add phenylalanine. For Nirenberg and Leder's experiment, they utilized short mRNA fragments that were synthesized and added to a mixture of ribosomes and tRNAs attached to amino acids. The reason why they used shorter mRNA fragments for this experiment was because they were just long enough to allow the ribosome to bind with the type of tRNA molecule that is complementary to the one codon and still remain detectable. The key step of this experiment was that they labeled one type of amino acid at a time and then put the mixture through a nucleopore filter. This filter allowed tRNAs that did not bind to any mRNA to pass through the filter, but it did not allow any of the ribosomes that were bound to mRNAs with the bound triplet attached to them to pass through the filter. So the sample was then tested for radioactivity. If there was radioactivity found in the sample that did not pass through the filter, the corresponding amino acid was then added, and it was concluded that the filter paper was radioactive if the trinucleotide codes for the radioactive amino acid. These results were major because it proved to be the Rosetta Stone of microbiology and led to many other discoveries. In this video,